we can go ahead and start back up again. So unfortunately, we learned that our panel is going to be a little bit smaller than anticipated at first. Unfortunately, Jonathan, Ava, and Shuo aren't going to be able to join us. So nevertheless, we will press on, and hopefully we can still get a little bit of good discussion. And we were, as planned, going to begin this panel, which, by the way, is going to focus on, how do we select, when we use Oxford Nanopore and other technologies for mapping modifications, what are the pros and cons of these different methods? What do researchers need to consider if they're trying to get into RNA modifications, mapping for certain applications or questions, and what should they know? What should they think about when they want to employ one method over the other? So those are kind of the things that we have on the docket to discuss on the panel, and we're going to begin by hearing a short talk from Dr. Jonas Korlach, who is from Pacific Biosciences, PacBio, that I think needs no introduction to this audience, and hear some of their latest and greatest methodologies as related to RNA sequencing. So we're very excited to hear this presentation, and then afterwards, like I said, we'll get into a smaller but hopefully productive panel discussion.
Great. Thank you so much, Kate. You can see my screen all right?
Yep, looks great.
Wonderful. I really appreciate the opportunity to tell you about some of the work that's been done by the community, mostly about PacBio sequencing for epitranscriptomics. And by way of introduction, there have been hundreds of papers using PacBio technology to resolve the complexity of the transcriptome in terms of isoform splicing and isoform diversity. So I only have 10 minutes, so it's going to be a pretty rapid fire and just a few examples. This is about 2 months ago, the new ENCODE transcript atlas, ENCODE 4, now finally has a transcript isoform resolution. This was done by 81 samples that were going through PacBio long read full-length transcript sequencing, or isoseq as we call it, and just a dramatic improvement in terms of the discovery. Importantly, more than one predominant transcript for almost 3/4 of all genes, which is in contrast to prior reports. So I should spend 10 minutes on this paper. If you haven't seen it, check it out. It's a tremendous resource. But so I just thought I'd highlight one vignette because mouse models were mentioned many times. So the researchers actually said the most surprising result to them in this paper was that the predominant isoform is for a given gene, so for orthologous gene pairs, the same genes in matching tissue, for more than half of the gene, the dominant isoform is different between mouse and human. And so that's pretty surprising. And the authors put a note of caution in terms of how we interpret mouse models, knockouts, and so forth. So check it out. In terms of isoseq and disease, there have been now many papers that signal a shift from gene expression analysis to transcript isoform expression analysis. And so this was one example in Alzheimer's disease, just one gene, ApoER2. This is the gene. These are the exons. Each column is an exon. Each row is a different isoform. Of course, there's massive complexity of the transcriptome in the brain. And now it's possible to, through the full-length sequencing, to associate these with regard to expression, isoform result expression, relative to whether they contribute to the disease, Alzheimer's, or not, relative to control. So region and disease-specific isoforms and the spatial and disease-specific regulation. And we see this in many other studies. This is aberrant splicing of this gene, but not that one, in sporadic Alzheimer's disease, isoform usage differences in schizophrenia, and so forth. With regard to single cells, and so, Kate, I know you're interested in the brain, so I chose this preprint, a novel cell-specific atlas of gene isoform expression that's reshaping our understanding of brain development. So again, this is a busy slide, lots of new information. For example, 27 megabases of human genome that is transcriptionally active but has no annotations to any gene code, GenCode gene model, and so forth. So these are the three main results: uncover novel isoform switches that are previously uncharacterized mechanisms in cellular identity of disease. Isoform-based cell clustering identifies previously uncharacterized cell states, so you get more clouds and more colors if you're isoform specifics. And then this is the kicker. reprioritizing thousands of rare de novo risk variants associated with neurodevelopmental disorders and revealing that risk genes are strongly associated with the number of unique isoforms observed per gene. So it's not gene expression, how much you make, but how complex and the number of unique isoforms. And so you mentioned earlier young and old brains. This is a PNAS paper from Gerald Chun looking at young, old, and also actually Down syndrome brains with isoform resolution, hundreds of thousands of unannotated transcripts. And here again, just one gene, this is BIN1. Here are the different cell types in the brain, and the color code relates to the different isoforms of just that one gene. And so you see that there's an enormous differences between the different cell types in terms of how these genes are spliced. There's been software that's been developed, as my last example, to uncover novel psychiatric disease mechanisms based on splicing, finding 12% of brain-expressed genes, that's almost 3,000 genes that are heritable only at the isoform level, and thereby uncovering previously hidden psychiatric disease mechanisms. And of course, in cancer, at least twice as many unique isoforms than other cell types, short-read RNA-seq, potentially leading to wrong biological conclusions because it fails to distinguish between gene and fusion expression. There's some nice examples in this paper. And generally, long-read sequencing providing a more complete picture of cancer-specific changes. The same is true for a very new isoform resolution, transcriptomic adult colorectal cancer, and so forth. So all of this new biological complexity that we see that is resolved with PacBio isoseq technology can now be basically put to question with regard to epitranscriptomics. And of course, 10 years ago, Kevin Shuo from Harvard showed that even very similar isoforms can have dramatically different RNA lifetimes and different decay rates and so forth. So in terms of isoseq for epitranscriptomics, Dr. Kate Meyer was one of the first to demonstrate that PacBio can be applied to this in a very powerful manner with DART-seq, applying it with PacBio sequencing. In addition to what she presented this morning, the full-length transcript nature of the sequencing allows you then to phase the different editing sites that are the signals for the modification, so it allows you to ask the question, if I have M6A on a 3' UTR, what is the probability that that same molecule, or what does that same molecule have some in the coding sequence or in the 5' UTR and so forth. And so this is the theme then is to basically apply everything that we heard in the morning session, all the different methods, and just use a different sequencing technology. And so it comes with the same advantages that you have with Illumina sequencing, the low input requirements, and the familiarity of the output and so forth. And so the papers that we saw, the presentation this morning with Glory, single cell DART-seq, and TAM-seq, all, to my knowledge, have not been utilized in conjunction with PacBio sequencing, but I think they're great opportunities because it's basically the same sample, but now not broken down into 150 base pair reads, but resolved in the full-length transcripts, and you get the contiguity in addition to the sequencing and the high accuracy and so forth. And then I had to leave for an hour, so maybe I missed this talk, but there's also for pseudouridines, a very similar conversion method, which now converts a pseudouridine into a deletion. Again, to my knowledge, this has not been applied to PacBio sequencing, but this would be as straightforward as all the other papers that I've listed, and you just use the full-length RNA and that goes into PacBio sequencing. Since I have to cover all the different angles of epigenetics and RNA in 10 minutes, so there are other opportunities or applications for epitranscriptomics. So for example, Jean Yeo had a Nature Methods paper, a very similar concept of now fusing an RNA binding protein to ApoBac1 and similarly converting this. And so here he shows that for different isoforms, so isoform A and isoform B, only isoform A is visited by that particular RNA binding protein. And some of you may have seen Gene's talk at the RNA Society meeting, where this is a very modular, system. So he's using two different RNA binding proteins and can resolve whether one goes before the other or whether they're co-localized and so forth. So this is a method to explore the epigenetic component of that. And then finally, in terms of RNA editing, which I think happens mostly tomorrow, was actually great timing because this paper just published 4 days ago by Ali Mortazavi and Dr. Shuo from UCLA to use PacBio sequencing for uncovering RNA editing sites. So check it out if you haven't seen it yet. RNA editing patterns in single RNA molecule resolution, co-occurrence, just like we saw from Kate's paper on DART-seq, and detection of allele-specific RNA editing. So this is an example here where you have these in blue here are the heterozygous SNPs, so that allows you to group the transcripts from maternally expressed, paternally expressed, and then here is an editing site, and you see that it's predominantly only from one allele, but not the other. The paper also has some interesting additional insights into highly structured regions. So the epigenetics of RNA folding and so looking at this paper looks at both Alzheimer's and also normal cells, and then the adenosine deaminase protein causing these high structures, and then you have RT read-through, which causes this skipped region. So one major limitation, of course, with everything that I said was that previously the throughput of the sequencing system was not very large, and so it was not very cost effective, it was expensive, and it was slow, and you didn't get a lot of reads. And this recently changed with the new Revio system, which we announced in October of last year and started shipping in March. It has a 15 times throughput increase compared to the predecessor. So in 24 hours, in a day's sequencing time, you get 360 gigabases of highly accurate Q30 plus hi-fi reads. In addition, for the particular case for transcriptomics research, there's a method that we commercialized maybe 3 or 4 months ago, which is a method that was developed by the Broad Institute to concatenate multiple transcripts, because a transcript is only a KB or 2, but a hi-fi read can be 20 KB. So for single cell applications, we're stitching together 16 full-length transcripts, and then we're sequencing that in one molecule. This is now a fully commercialized kit that's available for both the Sequel II and the Revio. And with that, it is possible to now, from a single run, get over 100 million full-length cDNA reads that you could imagine with DART-seq, with Glory, and these other methods. That really gets you into the regions that the regime that you can get with Illumina sequencing, but now giving you that contiguity, giving you the phasing, giving you the poly-A tails and all that. And so because you can run 4 smart cells on a Revio simultaneously, it's almost 500 million full-length reads in a day on a 24-hour time frame, which represents about a 200-fold overall throughput increase compared to before. So I think my 10 minutes are up. We have publicly available Revio data sets for both single cell, we call it MAS-seq, and also bulk iso-seq, bulk samples on our website if you want to check out the data quality and the depth and so forth. And I'm really excited by the opportunities that are now before us to apply this higher throughput and the full-length nature of the transcripts to all the methods that were discussed in the morning. I should have mentioned at the beginning that we currently do not have a direct RNA sequencing offering, but I think nevertheless, I hope through these few slides, I've given you some of the, I think, great opportunities to now apply all the methods that were discussed in the morning to the high throughput, highly accurate PacBio sequencing. So thanks so much. I think this was hopefully about 10 minutes, and looking forward to the panel.
This was perfect. Thank you so much. Yeah, I couldn't agree more. I think there's so much that can be done with, the iso-seq system and with the long read sequencing, and hopefully we can get into some of that in the panel. So maybe we can have the panelists, and I believe Dr. Novoa is going to be able to join us for at least the first part. Let's see. There we go. I see Logan. Fantastic. Great. So yeah, thanks again to all of you. And as I said from the beginning, the goal of this panel is similar to the one this morning, just to have a good discussion about, the strengths and limitations of these different methods, and hopefully be able to kind of guide researchers, in their choice of what to use and when, and, where the field is going. I think, if you have limited time, maybe I can start with you. There's a lot of excitement over, nanopore sequencing for both the single molecule aspects as well as the modification aspects. We're obviously here to focus on the latter, but I think the former is just as important as well. I wonder if maybe, in your talk, you mentioned kind of some of the major limitations currently to nanopore technology. I wonder if maybe you can just give us your thoughts on what you think the biggest thing is that, the field has to overcome to make, nanopore sequencing of RNA mods, a more widely used, technique.
I think that the technology is now relatively ready to be used for everybody. I think that in general, like, it still has some kind of bad press. Sometimes when you speak with people, they still think like you're in 70% accuracy levels, and that is actually very much of the past, right? Like, already that would be the R7 generation of pores. So I think that in general, anytime that you make a shift, into a new technology, there's a learning curve, right? So for sure, like getting the first, but then it depends a lot on whether you are using nanopore sequencing and you're doing the libraries yourself, or you just need to, externalize it to a facility, and then you're just doing the data analysis, right? So if you want to dive at the same time, both into the wet lab and dry lab, there will be some challenges, right? Because there's, associated issues with the flow cells at the beginning won't work, or, you will know what is going on, or you will have issues. So maybe I would tackle one of the two things first, right? Either you go for the wet or the dry, maybe not simultaneously, but I think that really it's something that is now already reachable for any lab that is interested in trying it out.
Yeah. When we talked somewhat of, this morning with the different speakers about, basically our benchmarks, right? Which I think is just as important in some of the more biochemical methods that we talked about earlier in the day as it is for the direct RNA sequencing technologies, like nanopore. I wonder if we can hear yours and the other panelists' thoughts on where we need to go as a field, like how do we establish the right benchmarks, and I think a lot of that is standards, having the right standards. As well as in the case of, we can just use M6A as an example. Obviously, Ava, in your own data, you were showing how we can get incomplete knockouts that give us results that are unexpected. And, what do we do with that? Do we as a field need to move completely to in vitro transcribed RNA libraries? Obviously that can be nice, but it has its drawbacks too, right? Because you can't get specificity of certain modifications. So, what do you think is the best way to tackle that? Maybe Ava and Logan can chime in on that.
You're going to go Logan first?
Sure. Yeah. So, to kind of go from short read standards, things like the SERVs spike-ins or spike-in controls that are known concentrations, known isoform distributions, and then if those can be synthesized with specific modifications at known positions, those can be an amazing just spike-in control that you add to all of your sequencing experiments and can be used in a way to check that the methods are finding the correct modifications at the correct stoichiometry on the correct isoforms at the correct concentration. Because the direct RNA sequencing is kind of this multimodal sequencing technique where you get a lot of different information from the same experiment. And so if we can have a control that tests all of these different aspects that you can get out of the data, then that's a nice thing you can do as an internal control. Now, I recognize that synthesizing nucleotide polymers with specific modifications at specific positions at kb lengths is no easy task. And so I know that's a very, very tall order at this point in time. But moving in that direction would definitely be, I think, the way we should start, at least trying to think about how to make it possible.
Yeah, I couldn't agree more on both those fronts, that it's really going to be necessary, but at the same time, it's not going to be easy, nor will it be cheap. But I think we have to establish what the ideal standard is and then find a way to get there, as opposed to just compromising and saying, well, it's going to be too hard and settling, because that's not going to get us anywhere.
Yeah, I guess that one of the things that is like in general, like especially if you want to like do orthogonal comparisons of things, right? Then like, because most of the things are done in-house by different labs, right? Then you can't really compare it afterwards using something else, right? So for example, like Logan was mentioning some of these standards, right? And like we actually have tried some of our software on their standards because they've been very nice to put all those FAST5 files publicly available, right? But then, for example, we get some stoichiometric measurements, right? We don't get 100%. So the question is the standard modified at 100%, or is our software not predicting 100%? Because if for that specific standard, mass spec, for example, wasn't done, or some, like then it's because, for example, there was this paper also, like I think that it was a Shraga with the AC4C, his spike-ins actually of AC4C were like 50% modification based on the mass spec, right? So even like when you do synthetic constructs, they may not be 100% modified. So if you're trying to benchmark how quantitative your method is, and you're trying to use third-party data of whichever source it is, right? That isn't like in general a problem because we don't have like commercially available modified standards. We just have like what other researchers are doing as best as they can and releasing as much as they can, right? And I think that this is an issue because for companies, they're not really interested in generating them because it's expensive. It's not really like a big market. But then we really need to kind of maybe come up with a systematic way of making some kind of resource. I don't know how, of some synthetic oligos or whatever that is available to the community for doing improvements in their methods and cross benchmarkings.
Right. I agree completely. I mean, that would be the ideal. Yeah. Oh, go ahead.
What you're describing, and I apologize, I'm not so familiar with your field, but what you're describing is pretty similar about the state of affairs in the genome space about, 7 or 8 years ago. And what happened then is that the NIST government agency actually, started the Genome in the Bottle program. And that was very helpful for all the reference genomes and pangenomes and all the advances that we have now, because they made a giant batch of HT002 and was giving out these tubes that all had the same genome in it. And so I'm wondering whether you could, whether it might be worth talking to them, whether they want to do a transcriptome in a bottle, because that's kind of, I think similar to what you are describing that is needed. And then everyone has at least the same sort of control that they're working with.
Yeah, I think that's a great point. And I know that, that idea has been brought up by several in the RNA mods community of having some sort of, central system where we can develop standards that are used throughout the community. And, those are the physical standards, right? But there are also computational standards. And again, I know we've already mentioned this afternoon, but that's one thing that we learned in these talks, right? Is there's not just one way you can analyze, nanopore data. I'm curious to hear the panel's thoughts on how we move forward in that sense as a field, right? Like, how do we get to the point where we can have more consensus in terms of how we call base modifications, or how we, if it's not, a base call, or how we identify them from neural network, training data sets that we use. I'm just curious what your thoughts are. Are we still too early days where we need to have a lot of labs working on this to help identify the best method, or is there something that we should already be starting to come to some sort of, targeted consensus in terms of how we analyze these data?
Well, go ahead.
Go ahead.
From my point of view, and you mentioned this, Eva, orthogonal validation, right, is a key. So again, equivalent to the genome world, there were these benchmarks, right? Specific benchmarks from the ABRF consortium, from the FDA Precision Truth Challenge, and so forth. And they tried to get as many orthogonal data sets as they could. And from the day today, we've heard that there's at least 7 different methods to measure M6A, nanopore and non-nanopore based. And so I think and then the Genome in the Bottle created their benchmark variant call set, their reference ground truth call set, by rules of having at least two very different technology call the same thing, and creating confidence there too. So and again, I thought that was pretty successful.
Yeah. From my perspective, I think that at the moment, it basically, like, each method has its own biases. So I think that you also need to think about which is your biological question, and based on that, you will also maybe find a better method. So, for example, if you're interested in looking at very low abundance RNAs at this point, you need throughput, and maybe in this sense, nanopore might not be your best. Until you get a lot of coverage, or now we will see with the RNA 004 and promethylene and so on and so forth. But otherwise, like until now, if you're very interested in, let's say, very low abundant long non-coding RNAs, well, nanopore is not the choice that you should make, right? Because you will have issues with having, minimum amount of coverage, or the possibility of even capturing a read that is going to be modified because you don't pre-enrich for your modified counterpart, right? On the other hand, I am, my personal bias is that at some point it will be likely more quantitative, because there's less processing and less PCR steps and all these kind of things that do include biases. So I think that once throughput is not an issue, I do think that nanopore should be more quantitative, because you have much less processing, no PCR amplification, no RT, so all these steps that add biases in principle should affect the accuracy in terms of quantitative measurements a priori. So I guess that depending on the question, I would choose one or the other as well, not just which is the best, but which is the best for your question.
Sure, that makes sense.
Yeah, I'll echo Eva's point. There isn't one tool for every problem. You do have to, in some cases, mass spec is the right choice. In some cases, short read data. In some cases, PacBio. In some cases, nanopore. And you really want to tailor what tool you use to address your question.
Yeah, I agree. And I guess I was trying to get to at more the computational side of things. If everyone's using nanopore, we face this, with things like MeRIP-seq too. You can have a lot of different ways to call peaks, and that can lead to differences in data sets. And someone says, oh, this peak is slightly more enriched than this one, therefore these sites are dynamically regulated, and then another lab might take the same data and do a different peak calling algorithm and see no difference, or see the opposite effect, right? It strikes me that nanopore at this moment is still, again, maybe earlier days, but still suffering for some of those same limitations, and that there are so many different methods out there. I think it's important to just think about how do you overcome that, right? And how do you choose which method?
Yeah, so to date, as far as I'm aware of, there's about 20 software tools for detecting RNA modifications from direct RNA.
If there's just 20, then we're probably
and there was a benchmarking paper that just came out earlier this year comparing about 12 of them, and on some fairly standard metrics. And I think with more effort like that, that body of work, and maybe making a systematic, like maybe a Nextflow pipeline or SnakeMake pipeline or something like that, where you can systematically use every single tool at the same time on a single data set and get fairly precise, reproducible calls from a variety of data sets for every tool, we can start making more of these kind of software benchmarks in a more reliable and robust way.
Mm-hmm. Yeah, I completely agree. Eva, did you have any thoughts on the computational side or to add? Or
I guess like when in general, I think that also an issue is to compare things. It's not always that straightforward for everything. So for example, I don't know, you maybe want to use this tool, but then it's only works with reads mapped to the transcriptome, whereas so for example, actually like nanocompore typically works for reads mapped to transcriptome, but we always map to genome, for example, right? So then that becomes a problem as well, right? Because then not every tool is that directly comparable based on what you want to do, right? So that adds an additional layer of complication. We did try to do a bit of and we have there's this master of pores pipeline that we kind of did, where we have tried integrating different tools, not just ours, to actually try to make a bit of this effort, and we actually use it ourselves to see, if predictions are consistent. And what we see is that actually, like by doing more than one software, you somehow rule out those false positives that each individual tool kind of has, and the consistent sites tend to stand out in a robust manner. So like basically what Logan is suggesting, I think that it does increase the accuracy and the specificity of the method, right? But then like, again, you can only do this for certain tools, or, you know, like maybe for certain modifications, or because each thing, or in this case, you know, like if you want to map to transcriptome or you want to map to genome. So it's not that straightforward for everything, but I do think that in general, these kind of consensus approaches can increase the accuracy of the results.
Sure, I completely agree. I wonder too if you guys can maybe comment for both Nanopore as well as PacBio, and this is partly from a question in the chat by Li Wu, referring to batch effects, and whether you can see, you know, whether you see a lot of variability from run to run, if that needs to be something that should be taken into account. And on that vein as well, you know, replicates, that's something that's been looked at in, you know, to stick with M6A, and for instance, Meyer mapping data is, you know, Sami Jeffrey and Chris Mason and others published a paper, you know, showing that most studies don't have enough replicates, and that you need like 7 or 8 replicates to actually get make meaningful conclusions. So I'm curious on both those questions, both kind of batch to batch effects, as well as how many, you know, replicates do we need to really nail down a well-defined epitranscriptome or transcriptome?
So there's been estimates that to reach saturation of a human transcriptome, you need about 45 to 50 million reads. And so if you're getting 2 to 4 million reads from a MinION on a really good day, 500,000 reads on a bad day, that's a lot of MinION sequencing from 002. And with a Promethean, 6 to 8 million reads is about what we're getting in 002. So to reach saturation, you need a lot of replicates and then pooling those data. And Promethean probably about 5 to 10, depending on your throughput. So with all of the software tools for finding modifications and for building transcriptomes, we're usually working with 5 million reads, 10 million reads at the high end. And so we're not quite reaching at least saturation of the transcriptome. With NanoCompor, we recommend 3 replicates for each, the reference and the test condition. But that's mostly to make sure you have enough coverage to do the statistics. And so yeah, replicates are important. And for reproducibility, the quality of the RNA makes a lot more difference than the sequencing itself. If you have the same RNA and you split it into 2 flow cells, the reproducibility between the 2 is quite similar. But RNA prep to RNA prep can be quite different. And so getting really good at that RNA processing step makes a big difference, at least in fragments.
That makes sense.
So for the PacBio side, you know, with the bulk mass signal, you get about 40 to 50 million full-length transcript reads. And what we found relative to saturation, that it depends on the tissue type, right? And so you know, so PBMCs and so forth, we see the saturation with one sequencing run and so forth. But for example, with muscle, you know, it's a lot less, and with brain, it's still going, right? So I think it's there's no one number. Like in genome, you have like 30x coverage, and that's a genome. For transcriptome, because the transcriptome complexity is so different in different tissue types, you know, that's something that needs to be considered. With regard to variability, you know, we're not recommending anything more than what you would do in terms of replicates based on the study or good academic practice. And I echo what Logan said with regard to the quality of the RNA. If you want to sequence full-length transcripts, you need to make sure that RNA is not degraded going in.
Any other thoughts?
Maybe just to briefly add in this, like, so at some point we were looking at what was the biggest source of variation in terms of batch effect, right? And like a kind of an agreement with what Logan was saying, the library, so the sequencing and independent flow cells is really not a big bias. The library prep itself, for nanopore at least, it's not a bias really. Like what really is the biggest bias actually is in the RNA extraction itself from time to time, because actually like if you do independent extractions not at the same time, that's going to be actually your largest source of variation. The fact that if you process the RNA and you do a different polyase selection in a different day, for example, right? That's going to be your largest source of variation more than the library prep. I think that this is quite different compared to other technologies like Illumina, where really you want to make sure that you process all your libraries simultaneously, because otherwise you do get a significant batch effect. That's what previous papers have shown. This in nanopore, at least, it's not a concern. The concern is more that if you did the preparation of your RNA in independent days, maybe you used slightly different bead selections, or you did a bit more harsh washing, and then your reads got more fragmented or something like this. So it's more the wet lab extraction of the RNA, the biggest source, than the sequencing or library prep itself.
Yeah.
At least it's in our hands.
Yeah, which is really important, because I think that's an often overlooked thing, right? Like so often people just assume, oh, well, there are HeLa cells here and HeLa cells there. We took the RNA, but those subtle differences can really add up. And I see there's a question from Yer and Kim in the chat asking if you recommend using public nanopore control samples to compare with in-house test samples. And I think that's, on this topic is important, right? Because if you have some public data set that you're comparing to, and this is true beyond just nanopore or PacBio technologies, right? That's something that we just kind of take for granted, that, oh, the RNA was probably good quality and this and that, but you don't know, right? And you can't necessarily compare one person's HeLa cell data set to your data set or hex cell data set or whatever it is, your brain or whatever. So I think these are really important things that all of us need to think about, regardless of the methods that we use. Jonas, I have a question about PacBio and the read accuracy. And I know once upon a time that was something that PacBio was really trying to improve, because there were a lot of kind of spurious, miscalled bases that was a big limitation to calling some of these. And you showed our DarkSeq data and other methods that like A to I mapping that all couple with IsoSeq, which as you beautifully laid out in your presentation, I think has a lot of power, right? These long read single molecule approaches can really tell us a lot about these transcripts that we just can't get from short read or other methods of sequencing. So I wonder if you can maybe comment on that, and if there's still an area that needs to be improved in that sense, or if things have improved.
So this is there was a dramatic shift around 2019. Now, IsoSeq data have always been circular consensus sequencing, and what that means is that we have we put in hairpins as on the ends of the molecule, and that makes it topologically circular, and you're sequencing the forward and the reverse strand of the same molecules multiple times over. So in fact, on the RNA side, IsoSeq reads have always been quite accurate. But now in 2019, the raw reads, the polymerase went long enough to do that for everything. And so that's where we introduced we branded them as hi-fi reads, which are typically 99.9% accurate or greater. With traditional IsoSeq reads, it's actually, because they're so short, it's actually Q50 and greater typically nowadays. And then when you concatenate with the MAS-Seq, we see again sort of Q30 plus. So Q35, Q40. So they're very, very accurate. And what that means is that you don't need UMIs in single cell applications, for example. And it's been shown by Hagentoven from Weill Cornell, for example, that the yield, the barcode calling yield is much higher than ONT, for example, and so forth. So that has changed, and since 2019, PacBio long read sequencing has been very accurate and has been the basis for not just the transcriptome atlases that I showed, but also on the genome side, the T2T was the base and so forth. But are there efforts to improve it? Yes, always. So we don't want to stop at Q30 or Q40. We want to give the community, and so the residual errors right now are mostly in long homonucleotide stretches. So our ability to call 8 Gs from 9 Gs or something like that, that's where the small residual error lies, and that's what we're working on. But in general, and this is the other thing with the bioinformatics tools that you mentioned, and the opportunity that I believe exists for DART-seq, for Glory, for the other methods. The data quality is the same or better than you get for Illumina data. And so all your tools should be readily available and applicable. The only difference is that you now get full-length transcripts. So I think there should be a very low activation barrier to applying and analyzing iso-seq data with the existing methods that now exist for Illumina.
Yeah, I completely agree. I think it's really kind of an untapped resource, right? Because we have all these methods now where we can, not all these methods. We have several methods where we can look at different RNA mods and, and as you talked about Gene Yao's data, for instance, using STAMP and TRIBE, and in my lab we've combined those to look at both TRIBE and STAMP for different RBPs simultaneously. Cool. I think that methods like that can be used in conjunction with PacBio to get information on individual isoform variants that might be differentially regulated by RBPs, and then throw in a DART-seq or a BID-seq or a Eta-I editing analysis, and now we can really start to uncover some exciting things. So I think it's very has a technology that has a lot of potential and currently is untapped.
Yeah, let's talk about that afterwards and see if we can do something.
Yeah, absolutely. And Eva, I think for Nanopore as well, I think it's very exciting to be using this in the context, as you showed, of uncovering codependencies of different modifications. I found those data to be really exciting. This is another thing that, in my opinion, at least has been, is kind of an untapped area that potentially could be, if we, focus on mRNAs and not just tRNAs too, could potentially be something that happens on a broader scale than any of us appreciate. So I wonder if you have thoughts of trying to incorporate more of that in your own research, looking at mRNAs and not just tRNAs.
Yeah, I guess that so far, the idea would be to do that once we have a NEP6A base caller a base caller for more than one modification, right? So I think that the next step is, once you have, for example, a base caller for inosine, for example, then one can start doing that question really at per read level, which is a very, very a great resolution to do that, right? Rather than like global correlations that otherwise can be done. In the case of tRNAs, it was that, like the presence of one completely kind of makes the other modification disappear. I don't think that that's going to be as evident with things like mRNAs, where modification stoichiometries are much lower, right? And that's why I think that for mRNAs, one needs per read resolution to really go down to the codependencies. And for that, we need base callers, I think, for different modifications.
Yeah, I couldn't agree more. And
so I'm sorry, but I do have to leave though. I'm sorry.
No problem.
But thank you very much for having me, and I'd be very happy to take any questions online afterwards.
Sure. Thanks very much. We appreciate everything that you've taught us today. Thank you.
Thank you, everybody. Bye. Take care.
So maybe in just the last couple minutes here that we have, we can hear Jonas and Logan's thoughts on, what to you is the most exciting thing in terms of, Logan, maybe nanopore and nanopore analysis methods for the field of RNA modifications, and not just calling modifications, but also being able to quantify them.
Yeah, so one non-nanopore thing that I'm really looking forward to is actually people using Glory with PacBio. I think that's going to be fabulous when people start doing that. And I think as Jonas said, coupling some of these Illumina techniques with PacBio will get you a lot of information that you couldn't get from the short read data. And I'm really looking forward to seeing those data set because I've been blown away by what they did with Glory. But on the nanopore side, I'm really curious to see either in ribosomal RNA, tRNA, non-coding RNA, and then I think to a lesser extent it happens in mRNA, multiple different types of modifications codependent on each other. And so if you have, is the mRNA processing or the modification processing, is there a temporal component to how the modifications are deposited on the strands?
Mm-hmm.
And is there a strict ordering or are they independent? And so I think that where nanopore can fit into this framework is focusing on many different modifications at once instead of one assay, one modification, and needing to do multiple assays and trying to piece together many experiments together, but trying to get multiple modification information in the same experiment. And so it's internally controlled as well.
Yeah, I completely agree. Sorry, go ahead.
I was finishing my thought.
Yeah, no, I completely agree with you there. I think that's a really exciting potential, down the road kind of thing that we can be exploring with this. And as Eva said, we need to have base colors for these different mods. We have to start with what we have the most training data for and, control data for, which of course is, M6A. But I think we really, as quickly as possible need to get to doing that with some of these other, at least more highly abundant modifications so that we can start to answer that question. Yeah.
Yeah, for sure.
Jonas, what excites you about this area? Oh, you're muted.
Can you hear me? Looking forward, I find it useful to look back a little bit. And over the last decade or so, we've seen tremendous advances using next generation sequencing and enormous discoveries and so forth, but also perhaps how much we're still missing in terms of the underlying complexity in the different omes, and therefore making our life hard, interpreting those signals into biology. And so what we've been seeing on the genome side with complete genomes, on the epigenome side, where we can now measure a 5-methylcytosine on the DNA as part of the sequencing. The Revio is a 5-base sequencer on DNA. Methods like FiberSeq that can give you long DNA resolved chromatin states. And then what I mentioned in this presentation on the IsoSeq side, there's one ome missing, which is the epitranscriptome. And I think these methods that you all have developed are very readily adaptable to PacBio sequencing. So I look forward to that. And then incorporating this particular data set into all the other omes that are now being generated that are comprehensive, that have long contiguity, that have haplotype phasing to get to allele specific information that you can't see with the short reads and so forth. So I think there's really enormous potential, and it's just there for the taking, basically. And then, as a company, we'll be committed and do the best what we can to support you all in that research.
Great. Yeah, I agree. Well, and thank you for what you've done thus far. And I agree. I mean, obviously, I'm a big fan of single cell omics. But I think on the heels of what you just said, we're getting there in that realm too, right? We have now the capability to look at more and more of these things at the single cell level, and I think the more ways we can do that with RNA modifications, of course, the better. And I think there's just a lot that we can uncover biologically when we do that. So I think very, very exciting future that we have for the field.
Yeah.
Great. Well, thank you to Jonas, Logan, and our other panelists and speakers this afternoon. I think this concludes the sessions for day one. We thank everyone for joining us, and we look forward to tomorrow, where we're going to start off the morning at 10:30, and we're going to begin with speakers who will be telling us about methods for looking at RNA modifications in tRNA. So we look forward to seeing everyone tomorrow. Thanks very much.
Thank you.
It's been a pleasure chatting with you all.
Yeah, pleasure.
Likewise.